General review of the immune system. The immune system distinguishes self from non-self and eliminates potentially dangerous non-self molecules and cells from the body. The immune system also has the ability to recognize and destroy abnormal cells that come from the host's tissues. Any molecule able to be recognized by the immune system is considered an AG. The skin, cornea and mucous membranes of the respiratory, gastrointestinal and genitourinary tracts form a physical barrier which is the body's first line of defense. Some of these barriers also include active immune functions. The outer keratinized epidermis. In the skin, keratinocytes secrete antimicrobial peptides, defensins, and sebaceous and sweat glands secrete microbial-inhibiting substances, e.g., lactic acid, fatty acids. In addition, many immune cells, e.g., mast cells, intraepithelial lymphocytes, AG-presenting Langerhans cells are present in the skin. Cornea. Neutrophils reach the cornea through the vessels of the limbus and kill microbes by phagocytosis. The mucous membrane of the respiratory tract, digestive tract and genitourinary tract. Mucus contains antimicrobial substances, such as lysozyme, lactoferrin and secretory IgA antibodies SIgA. The breakdown of anatomical barriers can trigger two types of response: innate, acquired. Several molecular components, e.g., complement, cytokines, acute phase reactants participate in innate and acquired immunity. Innate immunity. Innate natural immunity does not require prior exposure to antigen, i.e. immunological memory, to be fully effective. Thus, it can respond immediately to an invader. Innate immunity essentially recognizes molecular patterns that are widely distributed, rather than a specific antigen of a microorganism or a cell. Its components include phagocytic cells, e.g., neutrophils, monocytes, macrophages, polynuclear, innate lymphoid cells, e.g., natural killer NK cells, phagocytic cells, blood and tissue neutrophils, blood monocytes, tissue macrophages ingest and destroy invading AGs. Attack by phagocytic cells may be facilitated when AGs are coated with antibodies, produced as part of acquired immunity, or when complement proteins opsonize AGs. Polymorphonuclear leukocytes, neutrophils, eosinophils, basophils, mast cells and mononuclear ones, monocytes. Mast cell macrophages release inflammatory mediators. NK natural killer cells, which kill cells infected by viruses and certain tumor cells. Acquired immunity. Acquired adaptive immunity requires prior exposure to an antigen to be fully effective and thus takes time to develop after the initial encounter with a new invader. Thereafter, the response is quick. The system stores past and AG-specific exposures. Its components include less lymphocytes B, less lymphocytes T. Acquired immunity includes humoral immunity. Deriving from B-cell responses, B cells develop into plasma cells that secrete specific soluble anti-AG antibodies. Cell-mediated immunity, arising from certain T-cell responses. B and T cells work together to destroy invaders. Tissue AG-presenting cells are required to present AG to the bulk of T cells. Immune response. An effective immune defense requires the activation, regulation and suppression of the immune response. Activation. Immune system cells are activated when foreign AG is recognized by cell surface receptors. These cell surface receptors can be broadly specific, e.g., pattern-recognizing receptors such as toll-like, mannose, scavenger receptors on dendritic cells and other cells. Highly specific antibodies expressed on B lymphocytes or T-cell receptors expressed on T cells. Broad specificity receptors recognize molecular motifs associated with common microbial pathogens, such as gram-negative lipopolysaccharides, gram-positive peptidoglycans, bacterial flagellin, unmethylated cytosine guanosine dinucleotides CpG motifs, and viral RNA double-stranded. These receptors can also recognize molecules produced by stressed or infected human cells called damage-associated molecular patterns. Activation can also be observed when antibody-antigen, complement microorganism complexes bind to the surface of receptors in the crystallizable fragment Fc region of Ig Fc gamma R and for C3b and IC3b. Once recognized, an antigen-antibody complex or complement microorganism complex is internalized. Most organisms are killed after they have been phagocytosed, but others inhibit the killing ability of the phagocyte, e.g., Mycobacteria that have been included by a macrophage inhibit the killing ability of the cell. In such cases, T-cell-derived cytokines, particularly interferon gamma-IFN gamma, stimulate the phagocyte to produce more lytic enzymes and other microbicidal products, thereby enhancing its ability to destroy or sequester the organism.